For joining us today, Dr. Mohan.
My pleasure.
Pleasure to have you in post 2025. So I hope the journey has been good. The two-day journey has been good. So just to have an insight and walk us through your journey in oncology space and how you see it shaping over time.
All right. Thank you very much. It has been a wonderful experience for me because we've seen what's really happening, the cutting edge. How it comes into practice tomorrow is rather difficult to say right now, but unless you know what is happening, you'll never use it. So that's very exciting. So just to tell you my journey, I started my oncology last century, 1990. So what I will do is I will tell you about what's happened in lung cancer because that is a poster boy for targeted therapy, immunotherapy. So that time for us, it was enough if we knew it was non-small cell lung cancer, small cell lung cancer. But soon we realized that it was not just enough. We had drugs that was based on histology. So adenocarcinomas, we used to use pemetrexed. So that was a bit of the targeting for non-small cell lung cancer we had. But for the others, we were using non-pemetrexed based therapies. And then we were involved in a study called ISL, which is the first study for IRISA. At that point of time, not much was known about EGFR. They knew it was a tyrosine kinase inhibitor, but the knowledge about the exons, mutation, everything was not very clear. So this was a study which was done in 192 countries. India was a part of it. The drug studied was gefitinib in second line. That is, people who had progressed on first line chemotherapy, which is platinum based, were enrolled into this. And this study was a negative study. So it didn't show any benefit. But we had 110 patients from India. And we found that our patients were doing very well. Then the data from Southeast Asia also came in that their patients were doing well. Unfortunately, the Indian population was clubbed with Caucasians. So they didn't know much about how you. So we published this. Then they went back and saw that then we understood the EGFR mutation. Then they realized the EGFR mutations were much higher in the Southeast Asian population, almost 50-60% as compared to 4-8% in the Caucasians. So that's why that study was negative. But once they understood this, then the next study was IPASS, where they showed that this first study actually showed that patients of Asian origin and non-smokers were doing well. So this was a study which took us from, we always go from bench to bedside. This study from bedside to bench, we went back and understood the EGFR pathway. So now, because of that, we started looking at targeted therapies. And that's where targeted therapy first came in. And soon we understood so many more pathways. And today we have more than 50% of the pie of non-small cell lung cancer, which have slices per targeted therapy. I think that's a great thing. And non-small cell lung cancer, as you know, is a poster boy for targeted therapy today. So my days, it was just 3-4 chemotherapy drugs. From there, we have so many targeted therapies and immunotherapy. So that's been a wonderful journey. And what you have shown in the last 1.5 days is even more exciting. So as I showed there, my initial interest in CTCs started because we were trying to collaborate with the Basic Science Institute. So that's when I started working on CT-FEV and ctDNA. And I showed some of the data. But what you people are doing is fantastic. And I really hope that it comes into routine use rapidly. Because that is going to benefit a lot of patients. I know clinicians will want evidence, but unless you do it, where will you create evidence? So it need not have to be a clinical trial. It can be real world evidence. And today we know that real world evidence is as good as a clinical trial data. So that's where we stand, I think. And it's exciting times for everybody. And we are just the end users of what you do in the lab. So we really must be grateful to you for getting it across for us to use it for our patients. I agree, but it's more from your side to our side.
You ask the complex questions, and that's what motivates the scientists to go and find it.
Like you said, it's both ways, my teacher used to say, when you have a problem with pathology, just talk to the pathologist. Don't stay in your isolated silos. And I think it's become very important today. We have a lot of very good basic science institutes, but they're all working on their own in silos. They don't interact with clinicians. So I think it's important that we interact with each other. Exactly. Exactly. So that way, I think this was good because it got many of the clinicians to see what you were doing, and it opened up avenues for us to interact. I think that's very important. Thank you so much. Yeah, yeah. Go ahead.
You technology and AI with OneCell.ai. Where do you see it fitting in your protocol of everyday oncology routine?
Yeah, AI is slowly creeping in. So people are looking at it as a disruptive thing that they may lose their position, but I don't think that will happen. You will always be there. You should only use it as assisting you. So that's how I see it. And if that saves you time and some analytical thing, you can use that for something else. I think that's good. Thank you.